Hi everyone, welcome to Enzyme Logic classes. Today we'll be studying about the kinetics of enzymatic reaction. So to begin with, we have to follow the rate of an enzymatic reaction that follows Michaelis-Menten equation. So to determine the rate of reaction catalyzed by enzyme, we have to assume that the reaction is a simple reaction. The enzyme has a single catalytic site which binds to the substrate. There is no regulatory molecule attached to the enzyme, so that during reaction the complex will be consist of only enzyme and substrate, not any other molecule. The substrate concentration is higher to the enzyme concentration. An enzyme substrate complex is always formed during an enzymatic reaction. So that is the most important assumption we have. And the dissociation of enzyme and substrate complex to the enzyme and product is the rate limiting step in determining the speed of enzyme catalyzed reaction. Other conditions such as temperature, pressure, pH, which can affect the rate of reaction, remain unchanged. And the rate of reaction, wherever it is not mentioned, is always the initial rate. So it means that when we add the enzyme finally and substrate along with all other conditions, the start of the reaction, the starting speed of the reaction is known as the initial rate. Hence, today we will discuss how we can predict the rate of enzymatic reaction when the substrate concentration and enzyme concentration both are known. So, it is a bimolecular reaction, as the reaction happens only when there is a collision of substrate and enzyme. Enzyme and substrate complex must be formed, and the rate constants are mentioned as given in this equation. So when enzyme and substrate bind, the rate of this binding is K1, and after the ES complex is formed, it has two fates. It can either form the free enzyme and the products, or it has a chance to again form the free enzyme and the substrate. However, the formation of enzyme and product from ES complex is not a reversible reaction, as this step at the beginning of the reaction is always forward. Hence, we will see how the substrate concentration and the enzyme concentration affect the rate of reaction.